we spoke last time about how the body itself can communicate to the brainstem any type of stimulus that would warrant an emetic response. So you'll recall we talked about the important role that the vagus nerve plays in terms of its extensive innervation of many of your visceral organs in your thoracic cage or thoracic cavity and your abdominal cavity. We also talked about that it is possible that other sensory peripheral nerves could maybe play a role in that as well. So that is one way that you can communicate to the brain center. We also talked about that you can communicate to the brain center by toxins or by agents or emetic agents that could be in the plasma itself, making its way to the brain center, allowing the brain center to take a look at the contents of the plasma, so that can be communicated via blood. Then, of course, we talked about higher brain centers talking to the brainstem as well to effect an emetic event. So what I want to do now is take that brain center that we were referring to earlier, and let's expand this and see where some of the neurons are that coordinate all of this event. So what I'm showing you here is an oversimplified version, of course, of what really happens, but it's a way of organizing things so that we can understand the mechanisms at play. So I have a couple of boxes and squares here outlined for you. I drew some of this ahead of time to save some time. So what we're looking at here is I have a yellow little circle and a red box. So this yellow circle we are calling the vomit center. The vomit center is made of a collection of neurons, and its purpose is to receive information from other input, maybe higher brain centers, maybe from neuronal input from the body. It's going to take that signal, interpret it, and if an emetic event, if vomiting is warranted, then the vomit center is going to be responsible for coordinating all of the muscles that need to constrict or to relax in order to execute the event. You'll recall in an earlier lecture, we went over how everything has to work together. You have to have simultaneous contraction of the diaphragm and the abdominal muscles. You remember, the pyloric valve has to close, the lower esophageal valve or the cardiac valve has to open, the antrum of the stomach has to contract. All these things have to happen in order to carry out the emetic signal. So that doesn't happen by accident. Something has to call the shots, and what we're saying is that the vomit center is the one that coordinates that complex collection of muscle events in order to have a successful emetic event. So the two major components in the vomit center, one is going to be the nucleus tractus solitarius. The nucleus tractus solitarius is, we would consider a secondary sensory neuron or collection of them. And the nucleus tractus solitarius actually services several cranial nerves. So here it is down here. The nucleus tractus solitarius is going to service cranial nerve 7, which is facial. It's going to service cranial nerve 9, which is glossopharyngeal. And it's going to service cranial nerve 10. And cranial nerve 10 is the one we really want to emphasize at this lecture. So what you have is you have the input from the vagus nerve. And I'm going to go ahead and draw this out here. So I'm going to have fibers from the vagus nerve coming in, and it is going to be having a conversation with neurons on the nucleus tractus solitarius. Now, I'm saying that it is a secondary sensory neuron. The primary sensory neuron is the vagus nerve itself. In other words, primary being it is one big long nerve that is communicating, say, what is happening in my stomach all the way back up to the brainstem. So it's one neuron. So that's the primary sensory neuron. And so a secondary sensory neuron is going to be the neuron that receives information from the primary sensory neuron. And in this case, the primary sensory neuron is the vagus nerve. So it is going to be talking to the nucleus tractus solitarius. Now, another very important component of the vomit center is going to be the reticular formation. Now, the reticular formation, it's a broad term. You have reticular formation throughout your brainstem. And so the reticular formation is made of many, many named nuclei that are considered part of the reticular formation. I don't want to get into those individual names here. All I'm saying is that part of the reticular formation is involved in the vomit center. So what happens is that the neurons in the nucleus tractus solitarius, the secondary sensory neurons, it's going to receive that information, it's going to interpret that information, and then it's going to be having conversations with various neurons in the reticular formation, and as a result of that will become the coordination of the emetic event. So again, this is a terrible oversimplification, but it's the best that we're going to do here. So what's going to happen is the vomit center, again, nucleus tractus solitarius in the reticular formation, it is going to stimulate other neurons in the brainstem to coordinate all the muscle activities that have to happen for emesis. So I have some of them listed here. So what I'm going to get here is some of these neurons here. They're going to have to communicate, say, with the nucleus ambiguus. The nucleus ambiguus is a motor neuron. It's a nucleus, again, a collection of motor neurons in the brainstem, and those neurons will give rise to axons, and they will travel in the vagus nerve. And the target of the motor neurons associated with the nucleus ambiguus that's part of the vagus nerve are going to be skeletal muscles. So you will recall that when you're in the ejection phase and even in the dry heave phase, what's going to happen? What's going to happen is your glottis is going to close. Okay, so you're going to be adducting those vocal folds because you don't want to aspirate the vomit. So, and that's a reflex. So what causes the glottis to close? Well, it's going to be the skeletal muscles associated with the glottis, and it's being driven by motor neurons from the nucleus ambiguus. Also, we talked about how, say, during the retching phase, that upper esophageal sphincter has to be closed or has to be tight. And one of the major muscles that makes up the upper esophageal sphincter is going to be that cricopharyngeus muscle. Forms a little loop around there. So it is constantly constricted. Well, that muscle has to relax. Well, what causes that muscle to relax? Well, you're going to be dialing down the information from the nucleus ambiguus, and so that it's going to tell it to relax. Also, you're going to have elevation of the hyoid bone, elevation of the thyroid cartilage, so the larynx, literally pulling that open when we're talking about the actual ejection phase. So that involves even more neurons that are going to have to make that happen. Again, all coordinated by the vomit center. So that's going to have to go on, and at the same time, we're going to be having conversations with the dorsal motor nucleus of the vagus. The dorsal motor nucleus of the vagus, those are the motor neurons that are constituting the parasympathetic aspect of the vagus nerve. So you have this wide innervation of the vagus nerve, all these visceral organs throughout your thoracic cavity and most of your abdominal cavity. All of those are being innervated by neurons that live in the dorsal motor nucleus of the vagus. And so their job is to innervate all of those smooth muscles in the viscera itself. So remember we talked about the relaxation of the lower esophageal sphincter, or the cardiac sphincter as it's known, or the pyloric sphincter, or the contraction of the antrum. All of that is coordinated by the dorsal motor nucleus of the vagus. So that has to be talked to, and who talks to it? It's going to be neurons from the vomit center. You will also recall that a successful emetic event is going to involve a contraction of the diaphragm and at the same time contraction of the abdominal muscles, because we want to maximize the pressure in the abdominal cavity. And so in order to do that, that's going to involve the respiratory centers of the brainstem. They have to coordinate all of that. And so the respiratory centers have to be talked to from the vomit center. And then, of course, and this is not exhaustive, but you're going to have cranial nerve 12 being talked to or communicated with. That's my hypoglossal nerve, going to cause the tongue to protrude during ejection phase. I'm going to be having my cranial nerve 7, my facial nerve. That's going to be stimulating saliva, and also is going to be involved in elevation of soft palate. We have cranial nerve 5 involved. Again, that's going to be involved in several events that are important to emesis. Cranial nerve 5 is going to cause, one of the things it's going to cause my jaw to open up reflexively. I'm going to have an innervation of cranial nerve 9, the glossopharyngeal nerve. Again, that's going to be stimulating the parotid gland to make more saliva. So all of these things come together, and it is coordinated by this thing called the vomit center. And it's all being tripped off because you're getting some signal from the cranial nerve 10 or the vagus nerve. So you can quickly see how complex all of this gets. Before I move on, let me go ahead and make this point. The vomit center and the area postrema, or the central trigger zone, which is outlined in red, these are bilateral structures. So you have one on the left and you have one on the right. So I'm not wanting to imply that there's just one of these in the brain stem. These are both bilateral structures. So we have that. It is also possible that the vagus nerve will come over here and will somehow communicate to the central trigger zone. Now, whether it does it by one neuron or whether it is going to synapse onto another neuron and that neuron ends up talking to the area postrema neurons, we're not sure about that, but there's a lot of evidence to suggest that the vagus nerve can talk in some way to the central trigger zone without going through the vomit center. So there possibly is another route of communication there. There is a lot of crosstalk between the central trigger zone and the vomit center itself, and you would expect that because let us turn our attention to the central trigger zone. We will recall that one of the things it's responsible for is monitoring content in blood. So it is sampling what's in the plasma to see if there's anything in the plasma that would warrant an emetic event. And so I have a little blood vessel here. There are capillaries associated with it. Now let me say something about capillaries in brain and capillaries and spinal cord. Throughout most of the brain and the spinal cord, we have what is called a blood-brain barrier. The blood-brain barrier is there. It is literally a barrier because there are a lot of molecules in the plasma that are toxic to neurons and other brain cells. So if those molecules made it into the spinal cord or made it into the brain tissue, it would kill it. So you have to have that blood-brain barrier there because plasma is toxic to nervous tissue. The area postrema is different. There's something about those neurons there where they are not toxic to some of the bioactive molecules that are in the plasma. So not only do you not have a blood-brain barrier in the area postrema, those capillaries are incredibly leaky. And so these super leaky capillaries we call fenestrated capillaries. The word fenestrate has to do with window, so it literally is like the capillary is so leaky, it's like having a whole bunch of windows through which the plasma can leak out into the surrounding tissue. These fenestrated capillaries, again, they're responsible for communicating whatever's in the plasma to the central trigger zone. So it's going to pick up any type of emetic agents. Another thing, too, that's interesting is that the blood flow rate in these capillaries is very much slower than normal blood flow through other normal capillaries. And again, what that shows is that the purpose here of the interface of these fenestrated capillaries in the area postrema is to have a real slow bathing of the neurons relative to whatever's in the plasma, so that allowing these neurons to get a really good look of any possible emetic agents that are traveling in the plasma itself. So we've already spoken of this earlier. Two of the major emetic agents that could be found in plasma is going to be serotonin, and so I got my 5-HT here, and then substance P. So if you take an animal that can vomit and you put serotonin in their blood or raise the serotonin level in their blood, free serotonin, or if you raise the level of substance T in the plasma, the animal's going to vomit. So these are pretty powerful emetic agents that could cause emesis. Substance P, we spoke last time of it being associated with pain, and recall we had the spinal neurons, again, consciousness, your belly ache or your chest hurts, going and making a connection to a secondary sensory neuron in the spinal cord itself, and then that secondary sensory neuron goes up the cord and talks to the brainstem. So we're used to associate substance P with pain. There are other cells in the body that can make substance P. Substance P is released in many areas of the body, so it's not just associated within the spinal cord used as a neurotransmitter. What is really interesting is the neurons that we spoke of last time that are responsible for conscious sensation of what's going on in my stomach or in my chest. In other words, if I have a bellyache and now it hurts, again, the conscious awareness of the pain, that's not a vagus thing, that is a non-vagus thing. Well, those sensory neurons, they have the ability to release substance P into the GI tract. So not only are they functioning as sensory neurons, they can actually dump substance P into the GI tract. And if they dump it into the GI tract, then that means it gets into the blood, and if it gets into the blood, then it's going to be, again, eventually taken up to the CTZ, and again, could cause an emetic response. So those are kind of our two biggies. Another thing, too, is that the inflammation cells that are involved in our immune system, they can also release substance P. So if you have an inflamed membrane in your body cavity or maybe in your GI tract, part of the inflammation process may be to release substance P, goes into the blood, interacts with the central trigger zone, and again could cause emesis. So there are multiple ways that something going on in the GI tract can cause emesis. It could be, again, release of serotonin going through the vagus, or it could be release of other factors and being carried by the blood back into the area postrema. So lots of ways of getting there. And it's, of course, if I'm having a stimulation or the appropriate stimulation of the neurons associated with the area postrema, they've got to communicate that to the vomit center because it's the vomit center that's got to coordinate all the movement. And this is why, again, you have that crosstalk between the two. So this is what's going on here. And I just want to go ahead and draw this in carefully as well. And we'll go gray here. Not only from the peripheral nervous system do you have the vagus nerve, but I'm going to say other peripheral nerves. Maybe the splanchniks. They may also be coming in and having a conversation with the vomit center. Again, lots to be discovered on that. So I'm going to put a question mark here because, again, not quite sure, but they may be a potential feed into the vomit center as well. Again, that still obviously needs to be worked out. Okay, I think I'm going to end at this point, and then we're going to pick up on the next lecture, and I want to talk a little bit more about some of these receptors that we see here. I'll see you next time.